Listen-only mode.
Hello and welcome to today's webinar, the second in our webinar series, um, Smart Choices for Missing Data in Clinical Trials. I'm delighted to see all of you coming back again for the next webinar. My name is Andrew Grinnell, Senior Statistician here with Statistical Solutions. First, I just want to apologise for any inconvenience that may have been caused for the cancellation of last month's webinar, which is the rescheduled version of it. Apologies again for that. We've had some unforeseen technical difficulties. So today we're going to be talking about multiple imputation and the smart choices for multiple imputation in clinical trials. So just to outline the format, if you'd like to ask any questions at any time throughout the webinar, simply enter the text through the text box provided by the GoToMeeting window. I have my colleague Owen Murphy here as well, who'll be manning that, so he'll be passing any questions through and getting back to you as well throughout the webinar if you have any of those questions. Also, if you need to enlarge the GoToWebinar meeting or the GoToWebinar screen, simply use the zoom functions that are on the window there, and you can zoom in and out to your liking. And finally, if you can't hear me or understand me, please just send the message through the text box again, and we'll try and clear up any difficulties you have with the hearing or the seeing. So all right, so let's get started. So just to outline again, I mean, back in March there we had the first webinar of this series, overview of missing data causes of treatments in clinical trials, and there was great feedback from that. Thank you to everyone again who attended that. Today I'll be talking about the smart imputation choices for clinical trials, and we also have the third in this webinar series, which is successful strategies for sensitivity analysis, again for clinical trials, and that will be sometime next month, which will be announced soon. We'll give you plenty of notes for that. So today what I'm going to do is I'm just going to cover a brief review of last March's webinar on the causes and treatments of missing data in clinical trials. I'm going to give an overview of multiple imputation, the assumptions, how to approach it, and the various methodologies that we offer. And then also tackling multiple imputation, we'll do a worked example, and again we'll have time for questions and comments at the end. And please feel free to send them through at any time throughout the duration of the webinar. If I don't get to all these questions as well, like last time, I'll be sure to get back to every one of you individually. So let's get started. Just to review the last webinar in the series. Some summary points there. We talked about these regulatory agencies and a lot of the reports that they were writing in relation to missing data analysis, and also in terms of imputation. And we were saying that they all acknowledged the inevitable occurrence of missing data throughout the trial period. They felt that ignoring missing data violates the strict intention to treat principle. They all highlighted that missing data is a potential source of bias and loss of power and sample size. Missing data can affect the randomized sample that you've collected. Acceptable approaches will depend on the assumptions made and whether it is reasonable to make these assumptions, and again I'll go through that this time in more detail. And sensitivity analysis should also be used, and again that will be covered in detail in the next webinar next month. Also, we discussed how to minimize missing data in your trial, putting certain measures in place to try and reduce it as much as possible before you get to the end of the trial. Outlining the impact, as I said, with loss to power sample size and potential bias introduced. And finally, we ran through an example of how to explore missing data that does exist in your data set, such as the missing data pattern, margin plots, and overlaying imputed values. And again, we'll touch on that slightly again. We'll touch on that slightly here in this webinar again. So coming now to this month's webinar and what we're all about today, which is the smart choices for imputation in clinical trials. So I'm just going to give an overview here of multiple imputation for anyone who's not too familiar with it, and also we'll be going into a bit more detail too. So firstly, when you're looking at doing any imputation or missing data analysis of any sort, you really have to examine the underlying assumptions and the underlying approaches to missing data, such as the missing data mechanism and the missing data patterns that exist within your data once it's been collected. The missing data mechanisms that I'm talking about here, and which there's been quite a lot of documentation and quite a lot of discussion about, which you can find on the web, is the missing completely at random, missing at random, and missing not at random mechanisms. And then the data patterns that I'm talking about are when you kind of try and play with the data and re-alter the data so you can slot it into certain patterns, which helps you tackle which missing data points to impute first. And again, we'll go through that in more detail in a minute. So just to define first what the missing data mechanisms are. So this would almost be the kind of the first point when you're looking at your data once you've collected it. We're looking at missing completely at random, where the missingness is independent of any information that has been collected within the data set. Essentially what it turns out to be is just a smaller sample of the population that you are investigating. So it still is a random sample, it's just a smaller sample that you intended on, and it's still quite valid. It's just you may have some loss to power due to that loss in sample size. This can also be tested. There's a paper by Rod Little. I'll attach it to the YouTube video that goes out to the recording of this. And there's a paper by Little that he kind of explains how to go about investigating if it's missing completely random or is it missing at random. So that's an interesting paper to check out, and I'll put it up on the YouTube page. Then we have missing at random, and this is where the missingness can be explained by some of the information that you have collected, but is not related to the actual missingness itself. So you can explain how the missingness, you can model the missingness without requiring the missing information, essentially, and that's the key. And most of the imputation models that are out there, if not all, rely on both either missing at random or missing completely at random assumptions, and these can be investigated in different ways before approaching imputation. So there is some justification that you can give there before diving into using multiple imputation as a missing data method. And finally, there's missing not at random, where the missingness depends on the unseen observations themselves. So the missingness that has occurred in your data there, you actually require the information that is missing to explain the missingness itself, and this is what's called the non-ignorable missing data, and a different approach needs to be taken when going down that route. The next thing I said we talk about here is the missing data patterns, and we have two types of patterns, monotone and non-monotone. So I'm just going to briefly describe them and how it slots into approaching missing data analysis. So here we have a grid in front of you here where we have, it's essentially a square data set. We have 100 cases and we have 100 variables. These blue cells indicate a missing data point, and the white ones are observed. So at the moment here, it's quite scattered throughout the data set. There's no real order, and there's no real idea of which one do I impute first. If I am going to use imputation, which one do I go about filling in first? So there's a method then of reorganizing the rows and the columns, because in the end of the day, it doesn't matter which order they're in when you're doing analysis. So we reorder the rows and the columns so that we can bunch together as much of the missing data as possible. So as you can see here in the next image, you can see that we reordered the rows and the columns to now down the bottom right hand corner there. We've managed to put a lot of the missing data together into one clump, and then we have in the red cells and the blue cells there, then we have some outlying missing data points that can't be put into any cluster or any grouping. So these blue outlying missing data points, these are what we call non-monotone pattern. So these can't be grouped together or clustered together with any other missing data points to cause a bunch like there in the bottom right hand corner. And what Solus does in this case is it actually imputes those values first, so that you have as much information as possible to tackle the red missing data pattern there, where there is quite a lot of missings and such. And then this red corner here, that's what's referred to as the monotone missing data pattern. And essentially it's where every data point to the right is also has missing data associated with it. So this crops up a lot in longitudinal studies, where you might have an individual dropping out of a study for whatever reason, if it's withdrawal or coming off a treatment, and they are missing then for several time points within the data set, then a monotone pattern can be formed. So for example here, if we fill in the blue data points first, if you impute the blue data points first, then we have pretty much a full data set without the red filled in, and then that's when we tackle the red part. So we have as much information as possible before we tackle the monotone missing data points. Okay, now let's talk about multiple imputation here. So we have, in Solus, we offer 5 imputation methods. We have 2 model-based methods, 2 matching-based methods, and 1 combination of both. So I'll just briefly explain what they are first. So the model-based methods, they rely on some sort of a model to, you model the observed data, and you use this model to learn something about the missing patients or the missing cases in the data set, and you try and use that model then to impute reasonable values for the missing data points. So we have 2 here. Both of them are called, one is called predictive model-based method, and the other is called predictive mean matching. And essentially what we do here is we apply, for predictive model-based method, number 1 there, we apply a linear regression model to the observed data, and then we from the model we take the model values that are close to patients or individuals that have a missing data point. We take those data model values and we impute the model values for the missing data point for similar patients or similar cases. In the scenario for predictive mean matching, it's a similar approach, but we use a logistic regression model, apply that to the observed data, and then we find for a specific missing data point, or a patient with a missing data point, we highlight similar patients based on the model, and then we impute the actual observed data points close to the missing patient, and we use the actual observed data points for imputing instead of the model values. So in a lot of scenarios, this model-based method is preferred, as you're actually imputing observed values instead of model-based values, as the model has associated error with it. But sometimes, again, this linear regression model is perfectly fine for the scenario that you're in. We also then have two matching-based methods. There's propensity score matching and Malhoveness distance matching methods, where the propensity score matching method is we calculate the propensity for an individual to have a missing data point, and we use individuals or cases or patients with similar propensities, and we impute similar individual values based on similar propensities. Malhoveness distance works again in the same kind of way, where we compute the Malhoveness distance from a missing case to all the other observed cases in the data set, and we pull out the shortest Malhoveness distances, and we use them to impute the missing data point. As you can see here on the right, I have donor pools are used to create imputed values. So what does that mean? So we create a pool of values where they're either similar, or they have a small Malhoveness distance, or they have a similar propensity score, or they have a similar linear model value, or they've been highlighted as similar based on the predictive mean matching model, and we create a donor pool of 5, 10, 15 cases, and we randomly select using an approximate Bayesian bootstrap, randomly select with replacement from that pool to create the imputed values and create multiple data sets. Also, there's a seed used so that it orders the user to be able to replicate imputations. So when you set the seed, it can be replicated by colleagues, by regulatory agencies, and it's also advised that if you put the seed value into the protocol document, that it shows that it's set in stone, and when the regulatory agency gets your data, they can reproduce it immediately. I have a statement in bold at the bottom here, and sometimes people can overlook this interesting point. And essentially, it's the aim of imputation is to create the most accurate value to replace missing values in order to retain the information you've already observed. So we're not trying to come up with a real value where there is a missing one. What we're trying to do is we're trying to put a good enough value in there so that all the other information for that individual that you have collected, you can keep that in your analysis. So just say for contrasting, if you did a complete case analysis and you deleted out an individual with a missing data point, you've also deleted out information that you recorded, and you can't keep that in the analysis. So what we're trying to do with imputation is we're trying to come up with a good enough value to put in there so that we can utilize all the other information that you have collected on that individual. And then that complies with intention to treat principle, where just because they've dropped out, you're still including them in the study, and you're still including the information you've recorded about them in the study and in the analysis. So this next slide here outlines the difference between the matching and the model approaches to imputation. And this is where it comes into what I was referring to as the smart choices for imputation. It's because it's hard to talk about it in the abstract. It's a very case-by-case approach where a lot of input is needed by the actual researcher or the clinician or whoever is involved in the collection of the data. There's not just one approach to imputation, but that's why we believe in Solus, seeing as there's multiple approaches across both matching and model-based, that we can cover a lot of the scenarios that pop up, and there's always a go-to method in Solus that you can use when applying multiple imputation. So there's three main parameters that you want to be looking at when you're looking at matching and model-based methods. There's the level of nonlinearity. Obviously, for model-based methods such as linear regression and logistic regression, the performance, especially with the linear regression model, performance poor performance due to nonlinearity is high. But of course, the logistic regression is a bit more robust to that. Overlap between the predictor and the covariates. The regression or the model-based methods can be used as predictors, and then the correlation between the predictor and the covariates, this would not be affected much with the model-based approach. When we're looking at matching-based approaches, then level of nonlinearity is not an issue for the matching-based methods. Overlap, it works best with overlap between predictor and covariate values. It's always because you're looking for a match, you're looking for the best match for a missing data point. So if there's not any overlap or not any similar values, similar patients or cases within your data to match up with the missing data point, that's always an issue. But then you can also examine it differently in such a way that it might be an outlier from the sample, and there's other approaches to be taken there. And finally, the correlation between predictor and covariate values, the performance of the correlation it performs better when the correlation is high. Also, just to go back up one slide there, I want to just talk about this combination method then, and this is when this slots in, where it uses a combination of the model-based and matching-based methods to really bring the best of both those worlds together. And it's our newest method that we've included in Solus, and we've been getting some great results with it. And it's one of the newer methods out there in the field of multiple imputation. So, and again, if you have any questions there to run by me, keep sending them in, and we'll get around to them shortly. So I'm just going to run through now a short example through Solus of actually running an imputation example and going through the process of running multiple imputation. So I'll just go close out of this. I think I actually have some information on the data set here. So essentially what I tried to pull up here is just an example of a phase 2 trial. And the reason why I'm picking a phase 2 trial is because, and I have this sentence at the bottom of my screen, I don't know if you can see it there, is at the end of, you can have, when you have the end of phase meeting with your regulatory agency, be it the FDA or the EMA, you can have an open discussion with them about how you tackle the missing data, and seeing as the whole idea is that the phase 2 trial is just a much smaller version of the phase 3 where it gets expanded out, then if there's a good set of imputation models that work for the phase 2 data, it's good enough argument then to put it into protocol documentation for phase 3. So you can run it by the regulatory agencies at the end of phase 2 meeting and let them know, look, we've tried these and we've used exploratory missing data analysis such as the propensity score matching method, and we find it works quite well with the data that we have. We're willing to put this in as the missing data methodology and how to deal with our missing data at the end of phase 3. You can put that into protocol, and also with the seed values, as I said in Solus, with the seed values you can put those into the protocol documentation as well, and it just solidifies it and puts it more in cement what you're doing and how you're approaching it. So this example of a phase 2 trial, it's an intervention type of trial. It's a randomized longitudinal study, endpoint classified efficacy design. Purpose was for prevention. We've got a small trial of 50 patients and there were 11 incomplete records. These were due to both withdrawal and also dropout of the trial altogether. So I'll just close out of this and go into open up Solus now. So this is what Solus looks like. So I'm just going to open a data set here. That's one of the sample data sets that I can use. Also within Solus we've got a large range of import and export functionalities here. We have the Solus file format, BNDP, ASCII, CSV, Excel, and we also have a wide range of the statistical software packages available out there, such as JUMP, Minitab, SAS, several versions of SAS, both for Windows and Unix. We've got the SAS transport files, which are very important, the .xpt. We have S+, SPSS, Statistica, and Systat. So there's quite a large range of import and export functionalities within Solus, which can enable you to bring data in from another source, and also once you've finished doing your imputation or even doing any analysis, you can save that data as a specific file format and export it out of Solus into, say, SAS or SPSS or whatever software package you may be using. So I'm going to open up this data set here. And as you can see here, we've got 11 variables, we have 50 cases, we have symptom duration, age of the patient, and then we have 2 lots of measurements, baseline, 1 month after, 2 months after, and 3 months after baseline for both these measurements A and B. Now I tried to keep it as generic as possible because from talking with a lot of people after we sent out a request for any topics to be covered, there was quite a broad range of topics to be covered, and I felt trying to keep it as generic as possible, but in the clinical trial setting was probably the best way to approach it. But again, I'm happy to engage in any specific individual cases if you want to contact me after the webinar. That's no problem at all. So as I was saying, first, if we want to examine just the missing data pattern, so just go view missing data. So I'm going to click use all here. We have this simple drag and drop format in Solus, but also we have this use all button here. So I'm going to click that just to use all the variables in the data set. And then up pops this grid which shows us white cells for the present and blue cells for the missing data points. So as you can see here, we have blue cells here showing us where missing data points are, and white for all the observed. And I can also collapse this down to a summary grid. So I'll go view collapse. So now what this is showing here is we have actually 39 complete cases. We have 1 case with this particular missing data point. We have 1 case with this particular missing data pattern, and so on. So this is what the missing data looks like in its unaltered format. But I can also then rearrange this, as I was saying before, to highlight both monotone and non-monotone missing data patterns. So this is what it looks like now. So as you can see, we've rearranged both the rows and the columns to show this missing data pattern here. So this is the monotone missing data pattern here, and these are the unclustered non-monotone missing data. So we're going to tackle these first before we tackle these in terms of missing data imputation approach. So that's the missing data. So now when I actually want to run an imputation analysis, we also have a lot of features here in Solus where you can define variable attributes, do variable transformations, combine variables, and so on and so forth. And we also have many plotting features which I showed you the last day in terms of margin plotting, scatter plotting, and so on. So I'm going to jump straight into doing a multiple imputation example. I'm going to use this combination method, propensity score matching, predictive mean matching, Mahal-Novus distance combo method. So this is the imputation window that we can see now, and what we first start off with is a window showing us the variables that we want to impute, and then what we call the fixed covariates, so the ones that we want to use to define the imputation, or define the model, or variables that we want to use to set up the matching. So I want to use patients with similar symptoms, similar symptom duration, similar age, and similar baseline values, and I want to use those to define those 4 variables to define my imputation models and how I'm going to match and compare similar and different cases within the data set. And then these 6 variables, measure 1, 2, and 3, and measure B1, 2, and 3, I want to use those. Those are the incomplete variables in this data set, so I want to impute those. Now we also have then several tabs up along here at the top of the window. First one is the non-monotone pattern. So here we have the 6 variables that I want to impute. And what I can actually do is I can open up a specific variable and say how I want to define each individual variable when approaching monotone and non-monotone missing data imputation. So how do I want to set up how Solus tackles non-monotone and how it tackles monotone? I might want to leave them the same. Might want to slightly alter them depending on what variables are missing what. So, and we can similarly do the same for monotone. We can decide that at the monotone missing data point, I don't want to use age as a variable to impute non-monotone or monotone missing data patterns from measure A2 or something like that. So there's quite a lot of flexibility there for defining how to impute the missing data patterns for both monotone, non-monotone missing data for each individual variable. I briefly mentioned these before as well, these donor pools. So these are what we set up to create the actual values that we use to impute for a missing data point. And here we can see we can set up, the 5 closest data points, use the top 10% nearest observations, and so on. And this is just defining. You can personalize this donor pool definitions. And then finally, the advanced options here, you can set the main seed, you can set that value, record it, put it into your protocol documentation for phase 3. There's also setting tolerances for models and so on, and various different options here. So once you're happy with all the settings you set up, you can click OK, and it'll create your 5 data points, or 5 data sets that I asked it to create. There's a maximum of 50 in Solus. So now, that just happened very quickly there because it's a small data set and I'm just using it for demonstration purposes. But what we can see here now is we have a new window open, new data window open, and at the bottom here you can see that we have 5 tabs, one for each new data set that I've created. So now we have 5 data sets. If we want to look at, say, a scatter plot, so here we can see the relationship between 2 variables here. The blue triangles are the observed and the red are the imputed. And actually I can overlay then all 5 imputed data sets onto that one plot and see the distribution of each imputed value across all 5 data sets. Again, I showed that the last day. It's a nice feature to show. So you can see this imputation model is quite a nice fit for the trend that's happening in that relationship there. As you can see, the red data points are the monotone imputations, and the blue data points are the non-monotone. Again, I can hit save this data page so I can export it out in all these different file formats. So I can, again, if I want to do any auxiliary analysis, or if I want to bring it back into my own system, once I've done the imputations, you can bring these data sets back into whatever statistical software package you're using in its own file format. So that's quite a handy feature there as well. So I just noticed there we've gone over the 30 minutes there, so I think that's all I really wanted to show you anyway with multiple imputation in Solus. The next time we're going to talk more about sensitivity analysis. So once you do your post-imputation analysis, how do we approach then the sensitivities of each of the output from each analysis that you're doing? So just to round it off there then, the next webinar will be in June. We'll let you know, and we'll plenty of notice when that's going to be. It's going to be strategies for sensitivity analysis. Looking forward to that webinar, and I hope you all come along again. Also, if you're around, we have we're visiting several conferences over the next few months. So there's one, the Conference of Applied Statistics in Ireland is in Kildare this year, and that's next week. We'll be at that. There's also the SET and DIA meetings that are in Boston this year. We also have a presence at those if you want to come up and chat to us if you're there. So I'd like to thank you all again for your attendance, and I hope you found this beneficial, and I hope you found it interesting. We'll take a minute there to take a couple of questions and see what's been flying around, and I'll get back to you there just in 1 minute. So I just have time there to take one quick question there, and it's always a hot topic of discussion actually. So I'm just going to try and give my own interpretation on it. And it was a question about the threshold value for missingness. So is there a certain rule of thumb, or is there a certain threshold value where it's not advised to do imputation beyond this level of missingness in the data set? Some people say the level of missingness depends on the size of the data. Some people say if there's more than, I mean, from what I've seen myself, the rule of thumb seems to be 30% missingness. If there's more than 30%, you have to, there are various things you need to examine before you do any or approach any missing data analysis. But I would say again, it's a tricky one. It's a case by case situation really, because sometimes you have one or two variables in a data set might have a lot of missing data. So sometimes you exclude them, or you examine why they have a lot of missingness in them. Is it related to anything else? So there are a lot of things you need to check before you go about leaving any information out, essentially. But again, I'll contact you in relation to that topic as well myself, as I think we're just about kind of gone over time here. So finally, I just want to finish up again by saying thank you to everyone for attending again. I hope you all found it beneficial, and you got something more from it. And again, please feel free at any time to contact me if you have any questions on the topic. So look forward to seeing you next month for our next webinar in the series. Thank you.